Yes, okay, so Frida will be talking about Rondo, which is an interactive visual exploration tool for genome spatial connectivity. The floor is yours.
Yep, thanks. So we would like to, we think that we have a different take on visualizing IC data. So with IC data, we try to experimentally determine the 3D structure of chromosomes by cross-linking DNA that is close together in space and then cutting it with a restriction enzyme and then ligating it and then sequencing it. So what we do with the data is, at the very beginning, pretty standard. We do some data processing and significant calling, and we get tags and connections at different resolutions. And then we import that into Rondo, our tool, and we create what we call connectivity maps. So what are these? To better understand, this is the state of the art of how we visualize IC data. So we have a sequence that it's present on both sides of the matrix, and if a contact is present, then you have a dot. We think that this is not very straightforward for the untrained eye, and so we developed something slightly different, which is this. So now we see a connectivity map of the same data that I showed you before, and I hope that you agree with me that it's a little bit easier to understand. So what we do is to have the sequence that is wrapped around a circle, like a circus plot, and we cluster the data in, like over here, it's at 0.1 megabase resolution, and we have edges that are bundled together, and the thickness is this connection, and the strength, so the thickness is proportional, sorry, to the strength of the connection. And then, as you can see over here, you can add metadata about like cytobands, genes, and tags, so without crowding the visualization itself. So I was talking about plural, more than one map. So why is that? Because we think that we need to be able to zoom at different levels. So we need to be able to have an overview of what's going on at the chromosomal level without having too many things going on. And the same goes for drilling down up to the gene level. So at each resolution, we want to have like a minimum information, but the most like relevant information, a little bit like Google Maps, right? And then we want to let the user choose the different zoom level and navigate in this way. So what kind of data can you explore? At the moment, we have 15 published datasets and a novel dataset, and you can go on rondo.ws. It's online, it's a web tool, and as you can see, we have, or not very well, some information about the dataset itself, and upon hover, the quality is really bad. You have other information about the number of connections and the quality of these connections. That kind of rainbow madness that is going on outside, those are connection to other chromosomes, so trans connections, and as you can see here, I'm drilling down, and you have genes, and also you can filter connections by, sorry, going too fast for the video. So now I'm checking these connections that are trans connection, and over here, down here in purple, this one is a connection from a different area of the same chromosome. This is chromosome X. So you can filter connections. For example, here I'm filtering out everything that is trans, and then everything that is outside this view, which is super nice when you want to create a nice figure for a paper. But a feature that I really like about Rondo is the comparison between two data sets that you can do. So everything that is in bluish, it's for from this data set, and everything that it's in pink is from this data set, and everything that is in common is grayed out. We also have an integration with string in which you can see the proteins that are present in the area that you're looking, and you also get a nice kind of pop-up with the string network. So if I go a little bit further down, so another thing that I really like about Rondo, and I'm going to stop over here for a second, is that you can view different parts of a chromosome that are not adjacent in sequence at the same time. And that is really useful, especially when you want to use the search and you want to search for 3, 4 different genes or 3, 4 different chromosome locations, and you want to see them all together. And it's usually very, very difficult to do, and it's very easy to achieve over here in Rondo. So with this, I would like to thank, of course, Sean, the head of the lab, and particularly this work was the labor of love of the PhD student Kenny Sabir, and also Christian Stolte. And if you want to know more, check out my poster, is number B259. And thank you all, and thanks to the organizer for the opportunity. Thanks.